I'm going to hold it this way so that it looks just like what you've got in the lab manual, okay? No, not nervous, I just hate it. Okay, guys, ready? So we're going over, what figure is this? Figure 12.2 from the lab manual. So all of this that we're looking at here is a single muscle fiber. So what that means is that it is a single cell of skeletal muscle that's very, very long. So when you look at it, it's the diameter's in micrometers, but the length can be in centimeters. So it has multiple nuclei. So when you're looking at this, this will be the nucleus of this muscle cell or muscle fiber. This outer layer right here, this is going to be the plasma membrane that gets a special name, and it's called the sarcolemma. The skeletal muscle fiber then is specialized to contract. So inside, it's cytoplasm. So you've got a little bit of cytoplasm here in this yellow that you can see, and it's called sarcoplasm. It is going to be completely filled with these bundles of microtubules. Each one of these bundles that you see here, those are called the myofibrils. So the multiple myofibrils then are made up two principal contractile proteins. They're either thick filaments or thin filaments. The thick filaments are myosin filaments, and those are going to be thicker here. So what you see in this thicker lines, those are the thick filaments. And in these thinner lines, that is the actin protein, and those are going to be the thin filaments. Now, inside the skeletal muscle, the arrangement of these proteins is so perfect and so aligned that when you look at it under the microscope, it will give you this aspect of light bands, I bands, and dark bands, A bands. And what they are is simply when the light passes through, okay, in the area where the thick bands and the thin bands overlap, there it doesn't allow the light to pass through as easily, and that's the dark band, okay, so that's the A band. So when we look at the model, the A band actually runs from here to here, okay, and we're going to have then overlap between the thick and the thin filaments. So all of this here is the A band. When the muscle cell is relaxed, in the center of the A band, we'll have an area where we only have thick filaments and no overlapping. That's called the H zone within that A band. And within the H zone, we have these complexes of proteins that hold the thick filaments aligned to each other so that they're nice and straight. We're going to have this M lines. So you have M lines are these three. From here to here, you will have your H zone, okay? And from here to here, you will have your A band. Outside of the A band, when this muscle cell is relaxed, you'll have just thin filaments, and that's called the I band. The I band are anchored to another complex of proteins called the Z disc, and when we run from one Z disc to the next, that is the functional unit of this skeletal muscle, meaning the one that's going to contract. From Z disc to Z disc, that is called the sarcomere, okay? So when we look at this in a longitudinal section, that's how those areas of the sarcomere are going to look like. When we look at them in a cross section, so now we've cut them this way, okay, we're now going to see how we cut through the different types of filaments. Here, where we have only the thick filaments, this is going to be the H zone, okay? Where we have only thin filaments, this is the I band, okay? Where we have cross-linking between the thick filaments, this is going to be the M line, and where we see thick and thin filaments, this is going to be the outer edge of that A band. So this is the A band, okay? Outside of the sarcolemma, you're going to have that connective tissue layer that is going to isolate and wrap each individual muscle fiber, and this is called the endomysium. The sarcolemma is going to have specialized structures that are going to allow for the electricity to not only run along the surface of that plasma membrane or sarcolemma, but it's also going to have T tubules, these tubules that pass through. So you can see in the plasma membrane there's like holes. Those holes pass completely through from one end to the next. So it's completely lined with plasma membrane going along the inside. And these are going to be the T tubules. So when the electricity passes down the outside of that sarcolemma, it also goes deep into so that the inner myofibrils can also get the electrical signal that says now is the time to contract. The smooth endoplasmic reticulum is very specialized in the skeletal muscle cell, and it forms these complicated networks that you see in white, and it's called the sarcoplasmic reticulum. Very close to the T-tubule, the sarcoplasmic reticulum will form a widened sac. That widened sac is called the terminal cisternae. They're the terminal cisternae of the sarcoplasmic reticulum. Say that fast 3 times. So on each side of the T-tubule, you'll have 2 terminal cisternae. So 1, 2, 3. The 3 of those make a very important complex that's important for the physiology of how the muscle contracts, and that's called the triad. So when the electrical signal comes from the neuron that says now is the time to contract, it will pass down outside the outside of this plasma membrane, pass through the T-tubules, so here, and that will be the trigger signal to the terminal cisternae to release calcium from the storage inside that sarcoplasmic, and that initiates the whole sliding filament theory. These gray things are mitochondria, that we have to have abundant mitochondria for muscle contraction because we need the ATP. And I think I've said everything. Did I miss anything? All right. No, I don't think so. That's it.